Hello, everybody. My name is Michael Sturgis, and I will be hosting your webinar today. On behalf of Bio-Rad, I'd like to thank everybody for taking the time to attend. Before we get started, just a couple of housekeeping items. I want to let you know that this presentation will be recorded. You will receive a playback link that will allow you to replay the presentation if you wish, or share this information with one of your colleagues. If you require technical assistance during the call, go ahead and use the text chat feature that you see across the top of your screen. If you go ahead and you just see the red dot, follow that up. That is the text chat feature. Go ahead and send a message to CK Yang, and he will be happy to assist you. We will be taking questions at the conclusion of the presentation. If you wish to submit questions, you can do so at any time using this text chat feature. So today's presentation will be given by Dr. Richard Kraig. Dr. Richard Kraig is an authority on the role of glial cells in the brain. He balances research, teaching, as well as his clinical responsibilities at the University of Chicago Hospitals, where he is the William D. Mavey Professor in Neurosciences. Dr. Kraig earned his BA from Cornell University and a PhD in physiology and biophysics from the University of Iowa. And he earned his MD at New York University. He served as an intern at the University of Chicago, where he did his residency in neurology. And, he did his residency in neurology at the New York Hospital Cornell Medical Center, where he took his first faculty position before joining Chicago's faculty in 1988. Today he will discuss his work using the Bio-Plex suspension array system to measure key signaling protein changes to obtain data on critical node signaling sites involved in activity-dependent neuroprotection. Dr. Kraig.
Okay, thank you. So just one minor correction there of a very proud new affiliation I have. I actually attended my undergraduate time at Cornell College in Mount Vernon, Iowa. It's much older than Cornell University and the University of Chicago. And I just had the opportunity to join something called their Dimensions program. It's an outreach program for aging alumni to guide youngsters into the healthcare fields. And you'll see as we acknowledge people at the end that I've had two outstanding students from the college help me along with this work. So next is to test how good I am here at making this go. Here we go. So what I'd like to talk to you about today is I'm going to probably spend maybe a half to two-thirds of the time setting up the rationale for how I got to cytokine signaling in the brain and how development in about 2001 of the Bio-Plex system is sort of an ideal match to the scientific needs that we have. My basic thrust now in the laboratory is to be using multiplex signal analyses of innate cytokines and to determine how they impact brain function, and most importantly, how they impact brain function under normal circumstances. We're not so interested in disease, but in fact we're interested in the notion that cytokines, a principal reason for cytokines, is to activate genes, to create proteins, which in a conglomerate change the phenotype of the organ structure it's operating in. And I'll explain that more as we go, because this is key. They do this, we believe, and others have established this before us. We're going to embellish it quite a bit, that intrinsic neural activity itself has an immune subliminal signaling system that consists of innate cytokines as well as other immunomolecules that are responsible for the so-called ischemic tolerance, or as we would refer to it, intrinsic neuroprotection from neural activity. So in essence, I'm going to show you results that we are finally taking from abstract form to papers this year to show how cytokines can interact with neural activity to create neuroprotection. But of course, cytokines are a complex interactive signaling system, so the only way this is going to be possible is with Bio-Plex-like systems. Well, first, a little clinical background that I like to chime in with, and I'll show you why this comes to be important. There's a nice article by Chen in Neurology in 2005, and the basic thrust of it is that increased physical activity lowers the risk of Parkinson's disease, or those predisposed to Parkinson's disease, anywhere from early adulthood, middle adulthood, to later adulthood. So physical activity can strengthen brain to lessen the decline in a neurodegenerative disease. I'm going to come and show you that that applies to Alzheimer's, stroke, etc. And in fact, an Alzheimer's study found the same thing, that leisure time physical activity, anywhere from middle life on, decreased the rate of decline in Alzheimer's disease. So the question becomes the following, or the point I want to make is the following. At this leisurely activity found a 50% reduction in dementia, found a 60% reduction in those prone to suffer Alzheimer's disease that had the ApoE4 positive gene. So the first, if you will, caveat of, I think, the thematic or fundamental direction we're headed toward is that the gene is not deterministic. It helps establish a phenotype, but the environment creates a phenotype, and the gene creates a propensity. So that brain function, if allowed, can improve brain or deteriorate brain, determined on the genetic propensity. So the question becomes, is exercise alone enough? And I would argue no, and so does animal science work. And there's a nice a caveat to that in Archives of Neurology in 2005, that when cognitive activity is taken into account with those who are more physically active, there is no difference, suggesting that physical activity alone is insufficient to explain the improvement in neuroprotection. So next becomes the question, if you're running or walking volitionally, are you thinking? And does the thinking really change the brain function to improve your ability to withstand disease? What this follows from is an article that actually appeared in a lay journal, The New Scientist. It was written by Silverman, and I thought the gist of it captured it very well. His article suggested that prior assumptions, that is, that one gene equals one protein equals one disease, that is, the gene is deterministic, is rather archaic. Instead, it predisposes to disease. And that is, the old notion of a gene encoding a protein, and others don't coding a protein at all, needs to be considered. In fact, new systems biology approaches that go to gene-gene interactions or protein-protein interactions, that is a systems biology approach to complex signaling, is what's necessary to understand how a genetic propensity can either take you to a disease or take you away from a disease. He goes on, and this is where I, in fact, first came to the notion that the gene may not be solely determined phenotype. In fact, he looks at the cancer literature to say this is already a good example of how exercise, antioxidants, environmental factors that can change genetic propensity to disease further to the likelihood of having cancer or further away from the likelihood of cancer. Well, the basic thrust of our laboratory is to begin to look at how the environment, that is, not only the milieu within the brain, but the milieu of the organism, can create environmental conditions via neural activity that will change the brain structure and function to prevent neurologic disease. So, if you will, it's an extension off of this notion first established in the cancer literature that says genes may give you a propensity. The money to understanding the signaling system is in the proteins, but in the protein gemisch or milieu of the environmental exposure. So we became captured by the notion that thinking may be the key element of neurologic preventive health. And there's an article that was co-authored by from Greenhouse Laboratory, and the first author Anderson, all the way back in GLIA in 1994, that showed that looked at exercise versus motor learning and glial structure in the cerebellum. And their paradigm was either acrobatic activity, that is balancing on a wire, versus just simply running on a treadmill. And what they found was an old piece of literature, or reconfirmed an old piece of literature, that exercise can increase angiogenesis in the brain, capillary density. But that motor learning was associated with not only an increased number of synapses, but astrocytes, which vitalize synapses. And you probably will recall that astrocytic size is one of the first of 3 determinants of reactive astrogliosis. So that gliosis, a criterion of inflammation, actually occurs with a benign disorder, that is motor learning. They found fatter astrocytes in the areas where there was learning, not just exercise. Well, we've extended this to some degree, and I'll explain that a little bit later, to a phenomenon called spreading depression. Spreading depression is always regarded as a pathological phenomenon, but the more we are researching it, the more we are finding that it has many concomitants that are consistent with learning. Indeed, spreading depression prevents learning, but something that is already learned or activated to the top of the signaling system ability to learn can't learn any better. And that's part of what spreading depression, I think, is doing. Nonetheless, for our notions here, and perhaps you can ask questions at the end, the utility for spreading depression to us is that it is a benign perturbation of the tissue. It is ubiquitous through the tissue, so tissue sampling becomes very easy. And importantly for our slide here, it not only has astrocytic size increase, but microglial size increase, both of which are consistent with inflammation. So for now 10 to 16 years, the notion has been there that structural inflammatory changes of brain need not be associated with disease, but may in fact be associated with neuroprotection. This now, we're going to start moving to thinking per se. And we've copied many of the things, actually from a really wonderful review in Nature Review from Fred Gage's laboratory by Van Praag, who said most of what I'm showing you on this slide. The modern concept of environmental enrichment that I'm going to present to you in a moment really came from Hebb in 1949. And Hebb thought of it, but Rosenzweig and his colleagues ever since the 60s through 2000 have put this to a testable format, that is, an environmental enrichment paradigm to note that animals that are given increased social, intellectual, and volitional exercise opportunities learn. So this is a cage where we would typically house mice. A mouse cage is on the order of a cage that I'm starring here for you that looks like that, or maybe a quarter of a cage perhaps, and you put 7 mice in there. If instead you give them plenty of room to run, tunnels changed once a week, this is a food bowl, but it actually turned out to be a buddy bowl, a small dog water bowl that the animals kicked the food out of and socialized in so that there was physical contact, and a running wheel. And after 30 days, this was markedly protective. This first came to our attention in terms of epilepsy by an article in Nature Medicine in 1999. Young et al. Exposed animals to an environmentally enriched paradigm, I believe for a month, and found that there was a significant reduction in seizure frequency and, as I recall, seizure death from canic acid. That will be the paradigm I'm going to show you next. So we first tried to put this paradigm up online and see what would happen. Now, the purpose of our switching to this type of paradigm, and including it with spreading depression, which we study in a dish, is that this is a behaving animal in a normal environment that has increased thinking. So no electrodes in the brain to stimulate it, so it approaches its most physiologic state. And that's going to be key to doing cytokine analysis, as I'll show you, which are on the order of femtomolar signals in physiologic states. Well, the Bio-Rad system and that type of multiplex analysis is clearly capable of multiplex femtomolar signaling, so it's well suited to this. But if you shake my cultures, if you shake this cage, you can have relatively prompt and rather robust changes in cytokines, corticosterone, etc., that are going to confound your signaling. So an article I'll refer to later that we put out in Journal of Neuroscience Methods, first using the Bio-Plex, deals with some of that, how to handle the animal or the culture beforehand. Well, let me show you what we see. And this parallels what Young et al. Found, and that is that after canic acid injury, the white dots here show fluorojade staining of the caudal hippocampus. So CA3 selective neuronal vulnerability is lost here as I track along. That's CA3. This is fluorojade staining. It's inverse pseudo color just to bring out the red for you. Green doesn't show so well. And what we found was that after a month in environmental enrichment, and then 5 hours of canic acid, 5 hours after canic acid injection, that the number of cells per section were 117 in the unenriched and down to 23 in the enriched animals. And there was a survival that reached 77% and a mortality a little bit better than 50% with canic acid injections in seizure-susceptible mice. We went on with that via Yaping Tang, a memory expert here at the university, to help us show that the caudal hippocampus was in fact learning. So our enriched animals had a significantly improved freeze response time, so that the contextual freezing showed that our animals learned. Well, learning, the animal, the sort of cellular equivalent to it, and I won't belabor this very long, is long-term potentiation. This has been argued over the decades, but now seems very well established by in vitro but correlative in vivo recordings to show that synaptic potentials are elevated in those animals that learn. Importantly, I think learning is associated with an increase in back-propagating action potentials, so that as you stimulate to develop specificity, you know that you have an associativity in the postsynaptic cell. Well, that associativity includes a back-propagating action potential, and when it does, you get an increase in excitatory postsynaptic potential. We're beginning to think that it's key. These are movies that I lifted off the internet from the CNRC in, oops, it won't go on there. I can't put my laser on that. But anyway, this is normal action potentials traveling through on the left-hand side. And then if the frequency of stimulation is high enough, once in a while, a back-propagating action potential will occur. Well, that occurs with action potential, millisecond signals. What we found in studying spreading depression here to the right was that a similar but very slow back-propagating signal occurred. Electrogenic back-propagating signal occurred, and we published this in the Journal of Neuroscience in 2005. So while action potentials here to the left associated with learning fire rapidly in milliseconds, it takes seconds for these signals to propagate to the left. Nonetheless, they are electrogenic, and it made us wonder, could this be the postsynaptic red cell that is communicating to the orange microglial cell to establish a cytokine signaling network based on neuronal activity? And that's one of our working hypotheses in the lab, to try to tie neural activity directly to cytokine function of microglia. Others, including Beatty, and a very wonderful article in 2002 in Science, tied TNF-alpha to AMPA receptor insertion into postsynaptic cells. There's an article this week I'm afraid I've forgotten the author, but it's in this week's Science, that's showing that endocytosis of AMPA receptors is associated with learning LTP done in the dish. Well, this was the stimulus, those back-propagating signals, for us to further look at cytokines. And we're looking at innate cytokines because they are always there, ready to be expressed. They're a highly conserved series of proteins, and they are expressed in a subliminal or picogram per mil or femtomolar fashion. And we already know certain things about them. TNF-alpha, for example, like many other substances, this quote actually came from a review by Dernagel on ischemic tolerance. And then he noted that as early as the 1500s, the alchemist noted a toxin brought below threshold can become a nutrient. And if you will, that's the second theme that I think is important, that acute high exposure to cytokines is going to be associated with disease, but low brain-dependent activation of cytokines looks to be associated with protection. So these are a series of pictures. Now, being an old ion electrode scientist, I wanted to give you this analogy. This is from Morrison, and Morrison is Scientific American. It's a book called The Powers of 10. And what's attractive about it is, I want to just show you that we'll show you where this winds up. This is a fellow sitting on the lakefront here in Chicago, and it starts there. But we're going to go to sodium, which started in the 50s with action potentials in the Hodgkin-Huxley equations, and moved to potassium, which is a 10-fold lower signal. And many people started looking at gene activation based on sodium, gene activation based on potassium, as you search through the 60s and 70s literature. Well, shortly after that, calcium electrodes were developed, and these came in at around 10 to the 7, 10 to the 8, but pH came a little bit after it. Well, you know, pH of the cell is held at 7, and there's strong evidence to say that eukaryotic cell protein synthesis and gene activation occurs with an alkalotic change in pH. So pH could be a key factor. And you know, calcium always was. But these signals are very high. The buffering is pretty high in cells, so that the transients are localized very sharply to regions. To get cell-cell interactions, it'd be much healthier to look at a paracrine signaling system so that you could coordinate cells to a regional network or make one cell have the other change. And this is a third reason, if you will, or fundamental point why we've chosen cytokines. The fourth or fifth point is going to be that they worked. But really, so these are spatial, concentration being 1 over distance. And you can see that femtomolar levels of cytokines make quite a difference between sodium, calcium, pH, and potassium. And again, cytokines' principal purpose is to activate genes to make proteins, which, because of the proteins made and the environment exposed, creates the phenotype. And that's the experimental game we're after. Well, back to the environmental enrichment. We know from Bio-Plex measurements that there is a significant increase in TNF-alpha after 30 days in the hippocampus, and there's probably even in the higher region, the caudal hippocampus, where our memory and injury is seated. There's a trend change that almost reaches the significance of IL-1 beta, and IL-1 beta is a downstream innate cytokine from TNF-alpha. But we're going to focus for the rest of our day here on TNF-alpha. Importantly, there's strong evidence in the literature, and I'll show you some, that low exposure to TNF-alpha, like that induced from neural activity, is protective. Furthermore, I am a strong advocate of sleep and sleep memory consolidation, and there's a nice caveat in the older literature, perhaps a decade ago here, that says there's a 250% circadian change in TNF-alpha as you go to sleep in hippocampus. And we think that's going to be a key for us to focus for the cellular and molecular processes involved in activity-induced neuroprotection. So back to one of my favorite preparations. We've now gone in a dish, and this is the hippocampal organ culture shown to you on the right, which faithfully represents the hippocampus for at least 8 to 10 weeks in the dish. It is a deafferented structure, but importantly, the microglia become quiescent. Astrocytes stop expressing vimentin, which is a reactive cytoskeletal protein. So in terms of the glia function, it is really quite normal. Our JCBF and metabolism paper shows that the cytokines expressed in a horse serum-based organotypic culture really resembles that in brain, normal rat, comparable age rat brain very well. Well, this is just a voltage dye movie off to the left here to give you some idea of what spreading depression does. It doesn't kill any cells, but it can fill the whole tissue. This is a stimulating electrode here at the bottom, these two little black dots. It starts right adjacent to it, and then it progresses across the pyramidal cell layer. And a couple days after it, 3 days after it, here's what happens. This is cytox staining, where we measure the degree of staining, that is cell death in the CA1 area. We maximize it the next day, and we make a ratio of injury over max. This is the amount of protection one sees. So normal injury would look about that, 60%. And after spreading depression of 1 day, 3 days, 7 days, you can see that I've halved the injury. So like all ischemic tolerance paradigms where LPS is used, cooling, hypoxia, etc. This is an intrinsic activation of brain that reduces brain injury by 50%. And there's two important caveats here, which we're finally going to put out the door. And that is that either 3 days of pre-treatment with TNF-alpha or removal of TNF-alpha signaling with soluble TNFR1 abrogates the protection completely, and as a matter of fact, makes it actually worse than it was before. On the other hand, pre-treatment of the tissue with TNF-alpha at levels found during spreading depression generates protection. So for the first time, we know that spreading depression is associated with a TNF-alpha dependent protection. Okay, this means that neuroprotection can occur via cytokines, and it tells us that they're situation, cell, and very likely counterpart cell specific, so that to begin to develop targeted gene therapies to ameliorate these types of diseases, we're going to need to be considering what cell type, when, how much, how long, and what correlated cell signaling changes happen to occur with it. We think this will be very applicable to preventive healthcare strategies, that is, educating people or empowering them about how physical activity, intellectual activity, and social activity has a sound physiologic basis for reducing brain injury. But importantly, it actually leads to where sleep is going to come in on this. It gives us measurable outcome parameters, measurable physiology to understand, to establish pharmacologic bases to get the job done if we can't do it just through environmental enrichment. What I'd like to spend the next 15, 20 minutes doing is showing you how we're going about this. Well, I've told you that TNF-alpha generated the neuroprotection. I've mentioned it briefly that it happens during environmental enrichment in the whole animal. We've gone to LTP models in the dish and shown that it works there. We've shown that spreading depression works, and we're putting that all together now and getting it out the door. Certainly, TNF-alpha reacts via the TNFR1 receptor and might react versus TNFR2. There continues to be arguments about how much that is expressed, if at all, in brain. But we've drawn this map to give you a notion of just as soon as you say TNF-alpha, TNF-alpha goes to P38, goes to NF-kappa B, comes back out again via a variety of signaling systems, and, for example, can go back. TNF-alpha can excite or increase the expression of TNFR1. TNF-alpha can change the expression of IL-6, interferon, receptors, agonists. So this is an interactive system. The only way I think we can begin to really understand such an interactive system that, remember, by our experimental design is set up to say we are simply interested in the innate cytokine family signaling system's ability to impact neuroprotection via neural activity. Even that sort of, relatively small focus is gigantic when you start including the key or principally tied signaling molecules. That almost certainly means you have to do multiplexed signaling measurements. It will mean that the analysis of this data will have to take advantage of computational strategies. Our strategy for acquiring the data is going to acquire it in 3 to 4 sets of data. That is, we're going to first look at ligands, we're going to look at receptors, we're going to look at downstream phosphoproteins that include cytosolic kinases and selected transcription factors. We're then going to focus to the cell types of origin, where we draw the brain first as a universal cell, as we have here, but then be cognizant of the fact that the brain really consists of heterogeneous cell types, including neurons, astrocytes, microglia, oligodendrocytes, and capillary endothelial cells. We think putting those all together is the only way to actually get to the syntax of the complex signaling responsible for cytokine-based neuroprotection via neuroactivity. So how are we doing it? Well, first, of course, comes to the basis of our talk today. The first papers we've put out simply use the Bio-Plex system. And we use the Bio-Plex system in a spreading depression format because spreading depression was large, stereotypic, and therefore pretty easily made signals. We did have very nice response. We had 2 picogram per milligram sensitivity, which is femtomolar in origin. We had a very wide dynamic range. We measured 9 cytokines in our first go-through. We'll focus that slightly more to the innates now. And our intra- and inter-assay variability now is probably down to 6 to 8%. This is the data we published for both serum and rat, tissue, brain, serum, and organotypic cultures in the Journal of Neuroscience Methods. The cultures were published in this paper in JCDF Metabolism. And these are spreading depression results. You'll notice TNF-alpha falls quickly. I'm going to explain that response to you too. But TNF-alpha, like many have thought, is an early, and some may say first responder. Well, within the 6-hour period, we found they all responded. So you'd ask, well, why not study them all? And there's where we selected out using a soluble receptor antagonist to remove TNF-alpha. And remember, the purpose of our work is to show that it's involved in protection. And at this juncture, we simply want to say that TNF-alpha is a signal that is essential for neuroprotection. We are very intrigued by Hollenbach's work from the 1970s on that suggests that adding IL-1 beta or removing it may be a downstream signal, or maybe another innate cytokine signal, which we believe is a downstream innate cytokine signal responsible for the neuroprotection. We did find, let me back up a second and show you. There's a drop in TNF-alpha here. Well, that TNF-alpha either went away or was taken away. And there's a very easy way for that to be taken away, and that is if TNFR1 signaling goes up like a rocket, either in the soluble receptor or the membrane-bound receptor, you'll find a drop in TNF-alpha. And that's what we think is happening, and we're pursuing it now in our signal analysis paradigms. This is just TNFR1 immunostaining normally, and then after NMDA injury. And you'll see that on the neurons here, although this isn't double labeling, that there's a dramatic increase in TNFR1 on neurons. There's probably astrocytes in the background here in addition, but within hours of creating a TNF-alpha signal, you're already changing the receptor for it. So not only do you change cytokine ligands, but you certainly change receptors. Our next gambit, which we're bringing to the Society of Neuroscience this spring and then putting the paper together this summer, is to start looking at phosphoproteins. There are several specific phosphoproteins we chose as natural ones to be downstream in innate cytokines. This was derived just by literature review and by looking through a text called Cytokine Reference by Oppenheim and Feldman, which is just an outstanding 2-volume set. And we drew this map for ourselves as well as that map previously. And measuring these 6 cytokines, or reading the text, you can help establish for yourself whether IL-1 increases or decreases cytokine signaling. You notice that TNF-alpha does nothing more than increase everybody. IL-6 decreases. Some do both, like so. So again, studying a single cytokine, I believe, is probably going to be fraught with inconclusive, confounding issues, if you will. Well, I'll tell you that the phosphoprotein signaling kits from Bio-Plex are very easy to handle and actually will generate data more quickly than the cytokine assays we did. So they're a lot of fun to work with. We're still yet fussing with ourselves how best to work with the data, and that's the computational analyses I'm not yet prepared to talk about. So this is just a graphic representation of an up-down kind of signal. And these are hours after treatment with NMDA injury to show you that there is a clear profile change in many to all of the signals. And then the question becomes, which cell type, when, how long, what other changes are associated with it to see which signaling systems are key for this downstream signaling to evoke other protein expression. And I want to refer you to, I'm sorry I've forgotten the article, but there's an article, I think in 2003, that helped demonstrate this. This was a genetically manipulated mouse that put the membrane-bound TNF-alpha. It was a TACE-based system that either put it on a neuron or an astrocyte. And what they found was that when the membrane-bound TNF-alpha was expressed on the neuron, that there was no inflammation. When they moved it over to the astrocyte, there was blood-brain barrier breakdown and inflammation. So that the cell type generating the cytokine or the situation generating it in within brain where the cells are heterogeneous is going to be key to deciphering what signaling matters. I don't think it'll be adequate just to put TNF-alpha on the brain or remove TNF-alpha and generate an answer. Under rescue conditions with no injury, most of the evidence says at least microglia generate the cytokines. These are simply reactive pictures to show you the baseline staining of cells, and then evidence to show that at least in certain conditions, interferon gamma can come out of a microglial cell, IL-1 beta, IL-1 alpha. Curiously enough, though many have published with it, we've gone through 5 to 6 to 8 staining paradigms for TNF-alpha and have not successfully or reproducibly stained microglia for TNF-alpha. Therefore, we rely more on assays, such as the next type of technology we're coupling to our measurements. To get it to cell specificity, we've decided to go with laser dissection microscopy. And we're finding good success with Leica system because we can carve out specific neurons, astrocytes, or microglia shown in the top, middle, and the bottom panel. And now with their triple slide system, we can actually in one slide carve, or in 3 slides, put on the microscope tabletop, carve these cells within 20 minutes. Our work, pilot work from about a year ago, showed that 50 neurons, 100 astrocytes, and 150 microglia gave us 10 copy number resolution. Well, we're certainly modifying this now with more recent technology available to be in the single cell PCR style, so that we expect to be carving 5 to 10 cells per sample of neurons, astrocyte, and microglia, and making measurements within 20 minutes of it. This is our first approach. Remember, that's PCR and it's not proteomic. We are attempting to move the Bio-Plex system to a proteomic-based system for cell-specific measurements. As you might expect, if you look back to an old text by McElwain, to say, how much DNA, how much RNA, et cetera, is in the brain, you can begin to guesstimate to yourself how many cells are going to be necessary or how much protein amplification might be necessary. It will be a feat, but there is technology out there to get this done. And the last sort of thing I want to remind you of is that, especially in the dish, there are now multiplexed ways to go after functional imaging. This is Bio-Rad's gene gun. And so far, although we've had some issues with inflammation, it's clearly capable of generating multiple GFP stained cells. And this is the red is a LDL microglial background staining to show you their ubiquitous distribution and pyramidal cells in the organotypic culture. You can actually see the gold bead, that yellow dot in the cells here. And this would allow functional analyses and cell-specific cell area carving based on the functional state of the region of brain you activated. So we think putting these things together is going to give us a good handle on a goal we want to achieve, and that is to really study how learning and memory, and that is sometimes exampled by spreading the pressure for us, via retrograde signaling, alters gene expression in the milieu, astrocytes and microglia, via cytokines. We think very strongly that this cytokine-based exposure resembles a phenomenon termed homeostatic synaptic plasticity. That is, when a system is perturbed off of a baseline, it adapts to respond to it, to either establish a previous steady state or an altered steady state that is conducive to subsequent action. I'm afraid the speed of our talk now, I'll do a disservice to the neuroscience authors at Brandeis and elsewhere who have been principal proponents of this notion. We're extending it, we hope, with good science to the cytokine behavior as a paracrine signaling system to also be a homeostatic signaling capacity for neural circuit function associated with neuroprotection. We're going to extend ourselves to receptors, ligands, phosphoproteins. We're certainly going to be at a cell-specific level. And then we're going to tackle, and we are tackling now, how to make sense of our multiplex proteomic measurements and functional measurements using a correct syntax. Well, we've gotten most of this done with a whole slew of friends. Phil Kunkel was in my laboratory for years. He's now at the University of Indiana. Ray Hulse was a technician in my laboratory, and he's now graced the lab by coming back as a graduate student. Marcia Kraig's been my senior tech for a whole long time. David White's our molecular biologist. Yaping Tang's in psychiatry. He's the memory expert. George Langan is the director of ARC and helped with the animal cage development. These are summer students. Mike, Nick, Judith, and Wade and McCallan are two Cornell students. Well, Charles Nicholson's my thesis advisor, and that Journal of Neuroscience 2005 paper was a current source density analysis paper that let us see for the first time how the currents of spreading depression, not in speed, but in movement, resemble those of learning. And of course, over the years, I've had a lot of help First from Jennifer Walker when she was here. Joe Fednitishin helped us with the setting up the Bio-Plex, and Dennis Bittner has always been a help to us in trying to push us along to new things. And Andy Lees from Leica and gave me that real neat movie of the piece of tissue dropping. And I'll stop there, and I think I'm ready for questions or however you'd like to proceed.
Okay, thank you very much, Dr. Kraig. We will go ahead and take questions. Please use the text chat function to submit your questions. We'll give everybody a moment to go ahead and write them up. Okay, Dr. Kraig, our first question comes from Justin, and Justin would like to know where he can learn more about spreading depression.
Well, so I won't spend all the time on this. So I did my thesis in spreading depression, and that was 31 years ago. Spreading depression. Let me give you just a couple caveats on spreading depression. Spreading depression is thought to be the event that occurs with migraine headache. So when a person's eyes go bad, it propagates across the cortex at about 3 millimeters a minute. It is the only thing in God's earth that goes that slow in the brain. For 60 years, it's been regarded as a pathologic thing. It is also found at the perimeter of a focal stroke. So the penumbra gets worse when it fires spreading depression. Spreading depression is immense metabolic physiologic load on the tissue. I've been very interested since Nedegard and Hansen in 1988 published a paper that said it never killed cells, but it makes microglia and astrocytes active. The latter two pieces of work are mine. When Charles and I did the current source density analysis paper, it's in March 2005 Journal of Neuroscience, we were struck by how the current's direction and style really resembled learning. Well, Charles is more of an electrophysiologic scientist than I. I'm the holistic more guy, and he was amazed I got the words in, but I think the analogy will hold up. You can learn about spreading depression in a really, really excellent review is by a guy named George Samjian, and this is in Physiologic Reviews, 2001. George is just a few years older than Charles and I, but we're the old-timers who extend from the electrophysiology days, which is the 60s, to the ion electrode days of the 70s, to the chemical days of the 80s, to the molecular biology days of the 90s. Now the new 21st century is going to be the cytokine days of spreading depression. People have used this as just a way to, if you want a positive signal, Charles used to always say, study spreading depression. It really makes very nice square wave signals that you can change in the brain and measure. Samjian's review is probably the best to go have a look at it. And here's from Wendy. How much
yes, Wendy. How much?
So we measured. Yeah. So if I don't respond, you go ahead and give them to me. Right? Am I good?
The presenter. Yeah, I have to read the questions since the people listening are not able to see the questions.
Hey, this is like a courtroom.
So, Wendy would like to know
Your
how much
Honor.
brain tissue you need to measure. Yes. Wendy wants to know how much brain tissue you need to measure cytokines, in particular for small changes. So, go ahead. Sorry.
Yeah. No, that's okay. This is cool. Not so much. The Bio-Rad directions had this stuff up like 500 milligrams per mil. We're down to I don't know. It'll be wrong off the top of my head. The JCBF and metabolism paper says at least as low as you can go, which is like 100, I think. And we measured it from 15 microliter samples. So let me tell you the following also. Organotypic culture is 60 micrograms of total protein, and that's total protein, not wet weight. It's a milligram in wet weight. It's 60 micrograms of total protein, and we can measure a third to a fifth of that organotypic culture and easily measure the cytokines. But let me remind you of something. When you're in culture, those cytokines, 50 in diffusion occurs microns and milliseconds, so that those cytokines, if they're not absorbed on the postsynaptic cell or the adjacent cell, are not so likely to stay locked in the tissue, and you're probably better off measuring it from the supernatant milieu in culture. In brain tissue in vivo, I would suggest that, my little calculator, if I said 60, 60 divided by 5, 12 micrograms, 15 micrograms, 20 micrograms for sure should be enough to measure a sample. And we put those samples down to about 15 microliters and measure successfully.
Okay. I will wait just a moment, see if there are any additional questions.
Yep.
So you're finding that the approach you're using now gives you a sufficient sensitivity.
Oh yeah.
For your,
it's real overkill. Yeah. There's a little caveat in the Journal of Science Methods paper by Holtz. We actually found diluting the serum, and others have too. Diluting the serum gave a cleaner signal.
Yeah, I've heard that reported. There's a matrix effect that seems to dampen the signal if you use the serum undiluted.
Yeah. In addition, filtering it seems to.
Filtering seems to work as well. Okay. We have another question. Can you specify in more detail how you will be studying the connection between cytokines and signal?
Yes. So if you ever PubMed me, you will see that I'm an aging electrophysiologist. I grew up in a whole animal, went to slice, went to intracellular ion electrodes, and then got molecular. I am very focused to the organotypic culture because it is such a good replicate of brain tissue in vivo. Cell-cell communication is virtually normal to my eye and brain. Accordingly, you can stimulate a single neuron and with correct imaging, sample microglia adjacent to that activated neuron. So we are going to do cell-selective stimulations and being able to identify the functional change in those cells and monitor and harvest nearby or adjacent or regional changes in the non-neuronal cell types. Maybe I can be one sentence further specific with that. That won't always be and shouldn't be, I think, patch clamp electrodes because I don't like jabbing the electrodes into the tissue. It'll be voltage dye, metabolic dye, and post hoc tissue sampling for molecular analyses. We will activate the tissue as physiologically and non-invasively as we can. Watch the tissue behave and harvest anatomically specified regions and cells for molecular analyses.
And Jessica wanted to know if there's anything that you're doing in particular as you prep your samples for cytokine analysis.
That's when I said way at the front with that environmental enrichment cage. Our organotypic cultures live on an air-water interface membrane. Everybody will be familiar with them. If you shake that tray or bounce it on the tabletop, you've just activated the microglia, and you might as well quit for the day. If you leave it out at room temperature for 20 minutes, you've just induced the best neuroprotection you can possibly see. Accordingly, if you're going to do measurements, they have to be in a physiologic chamber. We've worked very hard to have that. We actually have an incubator chamber not the $25,000 version. It's better than you can commercially buy and put on top of your inverted scope. We have a CO2 regulated humidified chamber where we can place patch clamp electrodes or sharp electrodes and do recordings, but we're very careful that that stay as normal as possible. We are careful that we feed cultures in a cyclical basis with the same person feeding them, with no mischief in between, no bouncing, no leaving. When a tray comes out for culture feeding, it goes back within moments to a minute or two. That sort of thing is key. I didn't mention, we found when we first did the animals that bringing up a cage of environmental enrichment animals was impossible to harvest with little variance. That is, if you try to sneak a mouse out of these tunnels, they're going to run like hell. So we decided to anesthetize the entire cage with halothane and then let them all drop out. And that's in the Journal of Neuroscience Methods paper too, so that we are systematically anesthetizing the animals, and that has cut down the variance in the cytokine analyses in both conditions. And that's what I have to say there.
Okay, excellent. Sounds very challenging to get reliable readings.
And let's see, you got another one?
All right. I'll pause just a moment, see if there are additional questions. Presently, I'm not seeing another question.
Is that the how do you? Yeah, I have. How do you take prep samples for cytokine analyses from the brain? We do them. Our organotypic cultures are washed in phosphate buffered saline, centrifuge down, saline taken off, stored at -80. Our brain samples are taken from acutely anesthetized animals, brains withdrawn, fresh tissue frozen for histologic laser dissection analyses, and a core of tissue, a piece of that, maybe the half a brain, placed in frozen in isopentane for Bio-Plex analyses. So it's fresh, frozen, washed, and stored in a vial at -80.
Okay. I don't know if anybody has any additional questions. We'll wait just a moment. Okay. Well, if there are no additional questions, on behalf of Bio-Rad, we'd very much like to thank Dr. Kraig for his presentation. Also like to acknowledge C.K. Yang, who was behind the scenes helping us a lot with the technical end of this presentation.
Absolutely.
If anybody would, CK is invaluable. If anybody listening would like to learn more about XMAP technology or the Bio-Plex system, please visit www.biorad.com/bio-plex. Again, this presentation has been recorded, and very soon you will be receiving a thank Dr. Kraig. Have a good afternoon, everyone.
All right. Thanks very much.